Hello folks, welcome to the wet lab stream, or as I previously called the WL module, but they're supposed to be interchangeable. I'm just going to use them interchangeably. But for this particular quick presentation, I just want to quickly walk you through what we plan to create for our material and present to you. Essentially this is a WL overview. Now, some of this material at the beginning was prepared by Kat and myself, and it's been carried on forward with Maris's help. So Maris and I will be the instructor for this course, and so feel free to ask us questions at any point in the comments below. So let's get started on this overview. For the wet lab stream, we're going to focus primarily on in vitro experimental approaches, as well as some DNA assembly content, and also some basic bioinformatics work. Now, for those of you who haven't been in a lab before, I provided a picture for you to kind of imagine what it looks like. So this is my lab that I shared with the IGEM Waterloo team. And so you can kind of see in the back there, that's where our office is. That's where we do our bioinformatics, our planning, a lot of our experimental design. And out here is our lab space, where you can kind of see some common pieces of equipment. You can kind of see our electrophoresis equipment, where we look at DNA. Our centrifuges are over here. Our thermocycler is over there. This is the main workbench we have. Our sink is here, our reagents. All of our pipette tip boxes and consumables are laid out over here. Speakers for us to blast music because only students from IGEM use this lab, so late at night we can blast music. Fun times. And also a microwave to microwave things in. And so the content we're going to be teaching is not going to be super in-depth. And that's because, as I said before in the intro lecture, we're focusing on bringing rookies into the synthetic biology field, bringing people who don't have any experience in this field into the whole vast interdisciplinarity of everything in this field. So we're going to be focusing mostly on the theoretical reasons for how some assembly methods work for DNA, for how some protein purification work might look like, for example, and focus on practical material. We won't dive into the deep nitty-gritty biochemistry of, for example, how ribosomes, how they bind to mRNA, and how you can modify the specific amino acids in the ribosomes, for example, to re-engineer to have different specificity, all sorts of nitty-gritty things. We're going to focus on developing intuition, learning the concepts, so that you can take this stuff onward and learn it yourself at a much more in-depth level. We will also include a couple of tutorials for online tools you'll need to work with DNA and amino acid sequences, some common databases and software such as BenchLink and Chimera and JALView to do your bioinformatics, to do your sequence management. And lastly, in a couple of months from the release of this video, we will be releasing videos on the theory behind analytical approaches to help us better understand how our gene circuits are behaving in the actual system itself. So here are some additional photos for you to kind of imagine what wet lab stream content will gear you to work with. Up here you can kind of see us preparing some LB media, Luria Britannia media. It's what E. coli likes to grow in and what we use. Over here we have some PCR tubes. In this case, we're preparing them to be put into the gel electrophoresis machine that you can see over here. This is some common equipment. I encourage you to kind of look at different lab spaces to imagine how you might use some of this content. And I want to give some quick advice about experimental work. So be aware that experimental work takes time to troubleshoot, and it's the whole reason why biotech startups can often take some time, some years, to get to a final product that works. So just have that in your head. And as we go through the content, I'll try my best to give some advice for how to do controls and some foresight, things to look ahead of, things to plan for, basically. And also, please sequence all of your final constructs. If you don't know what a final construct is, it's fine. We explain what it is in the next lecture following this one. But make sure to sequence all your DNA to make sure your DNA sequence is what you're actually intending to work with. And so, as I said before, this module is not a substitute for very in-depth knowledge that you might need to do very specific research for to do graduate work with. And so, what we focus in this wet lab stream is to build the rookie up so that they can be more intermediate, so that they know the jargon a little bit better, and they can kind of talk the talk of synthetic biology to interact with more senior people. So to learn more about the wet lab content that you might need to know for more specific purposes, in case you're doing more advanced research, we kind of recommend these materials to help you advance your knowledge beyond what SimB 0 is focused on. So the two textbooks I recommend that I use for my undergrad are Voet and Voet Biochemistry 4th edition and Bruce Albert's Molecular Biology of the Cell 6th edition. And so the biochemistry textbook will give you some very good molecular background for what's going on with these parts of the genetic circuits that you will learn about. And molecular biology of the cell gives you some good context behind how these parts will behave in systems, prokaryotic or eukaryotic, and how they might be implicated in disease, for example, or environmental topics. In terms of learning more about the actual protocols to do the experiments, I recommend these two textbooks or these two texts that are available online. I definitely recommend them, especially for the iGEM students in the wet lab who may not have many resources to consult. These are good places to invest your money in to share with all of your future members. And lastly, we focus on the theoretical part of different experiments. And so we don't have a lab component of this online SimB 0 lecture. So I encourage you to check out Jove, which has a lot of different videos for basic biology experiments, including molecular biology experiments you would be doing for synthetic biology experimental work. And lastly, for people who are a bit more advanced, two places to go to get more synthetic biology and more molecular biology techniques that are advanced is Nature Protocols and ACS Synthetic Biology. I recommend these two journals because they have good, very well described content. Okay, that sums up our overview of the wet lab stream. So if at any point you have questions, comments, or concerns, suggestions, please leave them down in the comments below. We do check our comments very frequently because we're new to YouTube and we're very conscientious of how we can improve our material. And again, we're teachers, learn to be teachers. So if at any point you have comments about how we teach, how I explain things, how Marius explains things, leave them in the comments below. So without further ado, enjoy, have fun. Good things take time. And so some of these lectures are kind of long, but they do have breakpoints in a lot of them. And please use the timestamps to skip forward if you want. So yes, stay fresh, stay safe. I'll see you on the challenge, and thanks for watching.